Bismillahirrahmanirrahim. Today we are going to learn about the neuron and neuroglia. Neuron is the basic functional unit of nervous system, while the neuroglia are the supporting cells which are present throughout the central nervous system and peripheral nervous system. There are various types of neurons and neuroglia throughout the central and peripheral nervous system. In the diagram, we can see that there are four basic types of neurons which are present in the central and peripheral nervous system. These neurons are unipolar neuron, bipolar neuron, multipolar neuron, and pseudo-unipolar neuron. I'm going to write it over here. Unipolar neuron, bipolar neuron, multipolar neuron, and pseudo-unipolar neuron. These are the four types of neurons which are present in the nervous system. Unipolar neuron. What is the unipolar neuron? Unipolar neuron, as in the diagram we can see, this is the unipolar neuron in which the cell body is connected with only one filament, with only one pole. This is the cell body, and this cell body is attached with only one filament. That may be the filament of dendrite or filament of axon, but the cell body is attached with only one filament. This is called unipolar neuron. This one is bipolar neuron, in which the cell body is attached with two filament or with two projection with two poles. So this is bipolar neuron. This is multipolar neuron, as the cell body is attached are connected with the numerous projection or numerous filaments. You can see that these are the dendrites, large number of dendrites, and this is the axon. So the cell body is attached with large number of, with more than two filaments. That is called multipolar neuron. And this is pseudo unipolar neuron, and this pseudo unipolar neuron also exists inside the peripheral nervous system. This pseudo unipolar neuron, this is actually connected with one pole, which is later on connected with which give rise to two branches, either to dendrite and axon. So the neuron in which the cell body is attached with one pole, and that one pole give rise to multiple branches. That is pseudo unipolar neuron. Then the central nervous system and peripheral nervous system, all of these can exist, but the unipolar neuron, this type of neuron, only mostly exists in the embryonic stages. Now coming toward this diagram. In this diagram, we can see that this is a typical neuron, which is multipolar neuron, is having some basic parts which we should learn in this lecture. One of the basic part is the soma or cell body. This is the cell body which contain the neuroplasm and other cell organelles inside this, and also a nucleus. So this is what this is. Cell body. I'm going to start it from new numbering. A typical neuron will consist of cell body or soma, dendrite, axon. This is the cell body. This is the cell body. These are the dendrites. And this is the axon. So the difference between the dendrite and axon is that the dendrite always bring information from the body uh, to the cell body. It bring information to the cell body. The dendrite bring information or bring electrical signal to the cell body, and the axon take away the signal from the cell body. The axon take away the signal from the cell body, while the dendrite bring the information to the cell body. It bring to the cell body, and the axon take away from the cell body. The second difference between the axon and dendrite is that the axon are tapered. The dendrites are always tapered. You can see over here that the diameter of the filament of the egg of the dendrites become narrow as it goes further. It become narrow. So this is tapered, while the diameter of the axon remain the same throughout its length. It remains the same. There is no change in the diameter of axon throughout its length. So the axon is always cylindrical and having the same diameter throughout its length, while the dendrites are always tapered, are always going narrow and narrow. Okay, the third difference between the dendrites and axon is that the axon do not have any small bodies over here. You can't see over here, but you can see over here the small bodies. These are the small bodies inside the dendrites. These bodies are called nissel bodies. Nissel bodies. So the nissel bodies are present inside the cell body, and the dendrites, the nissel bodies are present inside the dendrites and cell body. Nissel bodies. Nissel bodies are always present, while the axon do not have any nissel body. No nissel body. Nissel was the name of the scientist who discovered this body, and this nissel body represents the ribosomes, which are scattered throughout the cell body and dendrites. So if the dendrite and the axon is having the same length, so then you can differentiate between the axon and dendrite by the presence of what? By the presence of nissel bodies. The next one is this is the Schwann cell. This is the Schwann cell, and it has produced a myelin sheet around the axon. This is another Schwann cell, and this has produced a myelin sheet around the axon. So the myelin sheet is produced by the Schwann cell in the peripheral nervous system, and this myelin sheet, the area of the axon where the myelin sheet is available is present. That area is called internode. This area of the axon which is enwrapped by the myelin sheet is called internode. Internode is also one of the covered by myelin sheet, and myelin sheet is produced by the Schwann cell, inside the peripheral nervous system, and by the oligodendrocyte in the central nervous system. I'm going to explain that in the next slide. While the internode, this is the internode, while the area where there is no myelin sheet or Schwann cell, that area is uncovered, and that uncovered area is called node. So node is the uncovered area of the axon. Node is the uncovered area of the axon. You can also say that between two internode there is a node, or between two nodes there is an internode. So this node is called node of Ranvir. Node is called node of Ranvir. Ranvir was a scientist who belonged to France, a French scientist who discovered this area, this node for the first time. That's why this name after his name. Okay. Now coming toward the next slide. So in this we can see that this is a sensory neuron which bring information from the periphery to the spinal cord. This is a sensory neuron, and this is the cell body. This is the dendrite, because the dendrite always bring information to the cell body, and this is the axon, because the axon bring in information, take away information from the cell body to the to this area. So this neuron is having supporting cell. These are the Schwann cell which have produced the myelin sheet all around the. So in the peripheral nervous system, the neuroglia are actually, I'm going to discuss in detail about the neuroglia. Neuroglia are also called glial cells. And these are the supporting cells which are present throughout the central and peripheral nervous system, and these are non-conductive. So there are 4 basic types of neuroglia in the central nervous system and 2 basic types of neuroglia in the peripheral nervous system. So in the peripheral nervous system, there are 2 types of what? There are 2 types of neuroglia. Number 1, the Schwann cells. The Schwann cell produce myelin sheet all around the axon, at the internode section of the axon. The Schwann cell produce these are the Schwann cell. But you should learn that, you should keep in mind that that the myelin sheet is not produced by only one Schwann cell. Multiple number of Schwann cells combine together to make the myelin sheet over here. For example, this portion is made by one Schwann cell, and this portion is made by second Schwann cell, and this myelin sheet is provided by the third Schwann cell. The large number of Schwann cell give myelin sheet to a single neuron outside the central nervous system. The second one is satellite cell. The satellite cells are also the supporting cell which give insulation and support to the cell body. These red color cells are the Schwann cell, which are all around the cell body outside the central nervous system, and this provides support to the cell body. It provides nutrition, and it provides insulation, and it provides support to the cell body, and it protects cell body from various types of anomalies. Okay, now coming toward the central nervous system. In the central nervous system, these are various types of supporting cell inside the central nervous system. In the central nervous system, there are 4 basic types of uh, 4 types of what? 4 types of neuroglia. Number 1, oligodendrocyte, which is very important. Number 2, astrocyte. Number 3, microglia. And number 4, ependymal cells. So these are the 4 types of cells or supporting cells which are present inside the central nervous system, means inside the brain and spinal cord. Coming toward the oligodendrocyte is the cell just like the Schwann cell, because this provide myelin sheet to neuron, to myelin sheet to axon area of the neuron. You can see over here that the blue color represents the neuron. This is one neuron, this is another neuron. You should also learn that some neurons are also having projection from axon or axon like structure, which is somewhat 90 angle to the original axon, and that is called axon collateral. This is called axon collateral. It is called, I'm going to write it here, axon collateral. Axon collateral is also to provide information to the nearby neurons, but axon collateral is not present in each and every neuron. So some of the neuron also contain the axon collateral, but you should learn that you should keep in mind that this diagram represents the brain, and in the brain the outer portion of the brain consists of gray matter, while the inner portion of the brain consists of white matter. So the white matter and gray matter, the white matter contain large number of Schwann oligodendrocyte, and these oligodendrocyte give myelin sheath to multiple neuron. This should there's a big difference between Schwann cell and oligodendrocyte, because one oligodendrocyte give myelin sheath to thousands of axons. One oligodendrocyte give myelin sheath to thousands of neuron. You can see over here that this is what this is oligodendrocyte, and this oligodendrocyte has given myelin sheath to large number to this axon and this part of the axon, this part of the axon. This has also give large number of myelin sheath to all around the axons. So one oligodendrocyte give large number of myelin sheath to thousands of neurons. And unlike the Schwann cell only gave the myelin sheath to a specific area of a single axon. So this was the oligodendrocyte, which gave insulation, which gave the internode to the in the form of myelin sheath to the axon, which are in the gray matter, which are in the white matter of the central nervous system. The second one is astrocyte. This is astrocyte, and this is also astrocyte. Astrocyte are two types. One is which is present inside the white matter, and the other which is present inside the gray matter. The astrocyte which is present inside the white matter is fibrous astrocyte. This is fibrous astrocyte, and this astrocyte is present inside the gray matter. This is called protoplasmic astrocyte. Astrocyte are two types. Fibrous astrocyte and protoplasmic astrocyte. So this astrocyte, what is astrocyte meant for? What is the basic function of astrocyte? There are multiple functions of astrocyte. The very prominent function of the astrocyte is it maintains the position of each and every cell inside the central nervous system. It is attached to almost all the surrounding cells. This astrocyte is attached with almost all the surrounding cell, which maintain the position of the cell, and which do not let any cell to change its position. So it maintains the position of the cells inside the central nervous system. It maintains the normal harmony of the brain. So this was one of the function. Another function of the astrocyte is that it provide intact blood-brain barrier. Some of the branches of the astrocyte, for example, this is a blood vessel. And this blood vessel is also one of the branch of this astrocyte is connected with this blood vessel, with the endothelial cell of this blood vessel. So this astrocyte enwrapped enwrap all around the blood vessel, and provide a tiny slit to the blood-brain barrier. So it maintains the blood-brain barrier. So the blood-brain barrier is maintained by the astrocyte, by the branches of astrocyte. If there is no if there was no astrocyte inside the brain, then it will become very difficult to maintain the blood-brain barrier, because there will be large slit inside the endothelial cell, and various types of chemicals or various types of entities can pass that slit, are just like the normal capillaries inside the body. So this is different from normal capillaries, because this is also having an intact branches of astrocyte, so it maintains the blood-brain barrier also. So the third function of this astrocyte is it provide support to the oligodendrocyte also. It provides support to the oligodendrocyte. You can see over here that it provides support to oligodendrocyte in the term of nutrition, in the term of various other protections. And this astrocyte, one of the very important function of the astrocyte, which has been revealed nowadays, that the astrocyte can work as a stem cell for the neuron. If there is astrocyte inside the brain, the astrocyte can develop into a neuron. The astrocyte can develop into a neuron inside the brain. So this is one of the very good function of the astrocyte, that the astrocyte can be differentiated into neuron inside the central nervous system. So this is also one of the important function of the astrocyte. Now coming toward the next function, the next cell, the microglia. These are the microglia, small cells. This is also the microglia, small cell inside the central nervous system, which are not on its own position. It changes its position throughout its life inside the central nervous system. It goes from one place to another inside the central nervous system, because this work is the macrophage of the central nervous system. The microglia work as the macrophage of the central nervous system. It moves from one place to another in the search of pathogen. If it see any pathogen inside, when it come in contact with the pathogen, it engulfs the pathogen, and the size of microglia get larger, and it engulfs the pathogen, and phagocytize that pathogen. So the main function of the microglia is phagocytosis of the pathogen, just like what, just like the macrophage. So it works as the macrophage of CNS. It's protect from pathogens. Okay, the next one is ependymal cell. These are ependymal cells. This is one cell, this is another cell, this is another cell. The ependymal cells are only present in the ventricle inside the brain. You can you all of you know that the brain consists of some ventricles. um, there are 4 ventricles inside the brain, and also a spinal canal. So all the ventricle inside the brain are lined by the ependymal cells. All the ventricle inside the brain are lined with the ependymal cell. The ependymal cell give an epithelial covering, or give a lining to the to what to the ventricles of the brain. So for example, this is one of the ventricle. uh, at this position, and at this position there are 2 ventricles, and one is over here and one is over here. So I have drawn only one ventricle, which is also incomplete, but this ventricle is lined by what? This ventricle is lined by the ependymal cell. So these ependymal cells do not let the fluid to come in contact with the parts of the brain. So the ependymal cells are meant for the provision of lining or epithelial coating to the ventricles of the brain.